So as promised, yesterday I said that today I was going to give a more regulatory talk, so this is it. So we're going to be talking about the US FDA regulatory framework. I think that, we've been talking a lot about assays and things. I think that I'm going to take a step back and talk a little bit about some of the more basic things, and then try to guide us as to where these assays fit into that picture. So starting with the very basics, and please recognize that I come from the FDA, US FDA, and some of the rules are close enough, but not quite. So it becomes quite confusing. So at the FDA, things that are drugs, that are considered drugs, are assessed by the Center of Drugs Evaluation and Research, CDER, and they include small molecules, peptides, oligonucleotides, and proteins. Now, mind you, vaccines, allergens, cell and gene therapies, and some proteins that are blood-derived are evaluated in a different center called Center of Biologics. So that is confusing because you have the Center of Biologics that does not evaluate biologics, but it's just to keep you on your toes, okay? To continue to keep you on your toes, we all understand what small molecules are. We all understand what proteins are. But at the FDA, at some point, they made a decision, and this took, I can tell you, hours and hours of meetings, very high-level meetings, that what is going to distinguish a peptide from a protein? For the FDA, that limit is at 40 amino acids, regardless as to whether it was synthesized or it was made recombinant, or a mix of two, right? Hybrid product. That's totally different from what the EMA says, which just they consider peptides that are synthetic, different from ones that are recombinant. So just be aware as if you ever go to other markets that there's different definitions and different requirements. And in fact, for the FDA at least, different laws apply. So the small molecules, peptides, and oligonucleotides are regulated under what's called the Federal Food, Drug, and Cosmetic Act, whereas the biologics are regulated under the Public Health Service Act. And so for these guys, when you want to license, you go for an NDA, new drug application. For these guys, you've heard of a BLA, biologics license application. Now, for small molecules, there's really a lot of very well-established regulatory guidelines. For proteins, biologics, there's quite a big list of guidances. For peptides and nucleic acids, that's not so much so, right? There is the guidelines are still evolving. There's quite a bit of disparate agreement or disagreement, I should say, as to where things go, where limits go between the different agencies. So I would say that they're evolving at this point, is what I would call it. Now, for peptides, nucleic acids, and proteins, you do need what's called an immunogenicity risk assessment in your submission. Now, yesterday, we also said that the classical thinking for immunogenicity risk is that there's so many factors and their interactions are so complex that usually to understand, what kind of incidence of immunogenicity, what are the clinical consequences, you need a clinical trial, right? So usually, immunogenicity in the clinical sphere is assessed during phase 1, 2, 3, and sometimes phase 4. How does that look like? So if you go to the guidance, it's going to tell you that you're going to have to present a sampling strategy. You're going to say, okay, I'm going to have patients and I'm going to test them on day 1, day 4, whatever it is that you think is reasonable, and you're going to have to justify that. Right? So usually it is coordinated, the samples are coordinated with PK/PD, just so that they understand if there's any issues of immunogenicity impacting PK/PD and vice versa. And a usual schedule might be like baseline and maybe 1, 3, 6 months And maybe at the end of treatment, if it's a monoclonal antibody, you'll probably be asked to do some samples far after so that you remove the antibody from the equation. The early phases, the focus is absolutely on safety, right? As you progress, the focus shifts to include incidence and loss of efficacy. So there's a number of assays. If you look at this, and I apologize, it's small writing, it's what's in the guidance, but they have a screening assay, and if it's positive, you go to confirmatory, and if so, then you go to titrating, neutralizing, and so forth. Of all of those assays, the ones that's absolute is an absolute need is that screening assay, right? Whether you don't need neutralizing antibodies for all the products. Most of the times, if you have antibodies, they will ask you for titrating because they would like to know what level of antibodies. But that is sort of based on a way of thinking that involves thresholds, right? Levels that are up to a certain threshold considered positive, and under the threshold is considered negative. And once it's positive, you want to know how positive. There's other ways of looking at antibodies that, for example, signal to noise, where you need to do that extra step of the titrating. So there are alternatives. And the reason why I'm bringing this up is because a lot of these alternatives are really risk dependent. And what you want to make sure is that the agency understands what your strategy is, and that you explain it, and that you consider all the things that need to go into this so that you get the best design that you can, and you put it in your immunogenicity risk assessment. For example, depending on your risk, you may not need to test those samples from a phase 1. They may say, if there's no adverse events or anything, you may just collect samples just in case. And that might save you a lot of time and money. So it's just, again, or you may not need to do 10,000 patients in your phase 3. You may need it for other things, but maybe not for immunogenicity. So these are things that you want to consider, you want to think about, maybe with somebody who, to help you figure out what is the best course of action for your product. Now, I know that you've seen this. Other people, we all have our own little designs of how to illustrate the risk factors, right? So we've talked about patient and treatment-related factors, treatment, dose, route, regimen, mechanism of action. Important to understand the immune status of the patient. It's not the same patient with autoimmune disease that a patient with leukemia, right? Completely different immune status into which the drug is going. And then there's the product-related factors. And so you have the degree of existing tolerance. And people focus a lot on the homology to self. And I wanted to bring this up because I just was working with a client. And they were, they had high homology, 100% homology to the peptide that they were looking for. So they expected no immunogenicity, except that they had put a little carrier peptide in there that had high homology to a microbial peptide. And lo and behold, they had 100% immunogenicity. That was, if you take a minute to think about it, it's not surprising, but you need to think not just of homology to self, but also homology to whatever else is out there. It's just something to think about. So in addition to the existing tolerance and whether the product has immunomodulatory properties, there's the issue of potential impurities, right? So usually, when we talk about impurities of the API, of the active product ingredient, right? Or sometimes people call them product variants, usually you will need to understand variants that are above 0.1%. Depending on the agency, they might ask you different levels of understanding, but you will need to control and understand. And then there's the process-related impurities, aggregates, host cell proteins, contaminants, residual PAMs, which I might want to stress is different from sterility. People confuse sterility with the absence of microbial remnants. Those are two different things, and they should be considered and thought about differently. and of course, leachables. Now, for impurities, that's where one area, for example, where preclinical testing can really help with risk assessment and mitigation. So, yesterday, Nick presented about the economic value of actually testing your product rather than waiting to see if it works or fails. If you consider that, depending on the studies, people say up to 90%, it all depends on how you consider things and what you put in and out in numbers, but a high number of products fail, don't get to license, right? And that has driven companies to really use these NAMs to help select candidates, optimize target molecule, and inform clinical strategy. All of that is usually internal, and it's very useful. But currently, this information is usually not provided to the agencies. Now, that has some clear advantages. If you don't provide what you did, the agency cannot ask you, why did you decide to go with this molecule rather than the other molecule, which is, after all, a business decision, and that's perfectly fine. The disadvantage is that they cannot provide regulatory relief based on data that is not there. So it's not enough for you to say, we looked at the immunogenicity risk, we don't think there's any, we're fine, we don't need to test. That's not going to go very well. You're going to be asked to test, right? So you will not get relief unless you explain your results and where they came from. It's also, in my view, personal view, a lost opportunity. I think that the whole field advances a lot more if we're a bit more open about data and things that worked and things that didn't work. That's just my personal view. So inclusion of NAMS data in an immunogenicity risk assessment can help inform regulators of the immunogenicity risk, the steps that you've taken to mitigate it, and that may allow for some regulatory relief. So consider that when you're preparing your submission. So this immunogenicity risk assessment, where does it go? It is part of the integrated immunogenicity summary. It usually is put in sections 2.7 or 5.3.5.3 of the ECTD. This is just where it goes. It's very important it goes in the same place because then we can find it. It's a huge document. It's hard to find. So it contains a comprehensive evaluation of product, treatment regimen, patient-related factors, and everything that may influence the potential for immunogenicity of the product and what is the action plan to control it, right? It justifies internally, it helps you determine how you're going to study that product, and to the agency, what are the non-clinical and clinical strategies that you're using to assess it, right? So when you have an NME, and by the way, both new molecular entities and follow-ons require these assessment, but when you have a new molecular entity, your focus at the beginning should be, okay, I know that you think that this is not going to be immunogenic, but if it were, how would it manifest itself? What would happen? Be clear about what are the potential consequences and why you think that it could not happen and so forth. So if you're clear about it, that helps understand what the risk is, and it helps the agency understand what the risk is. And then you also want to include all the preclinical things that you did to assess that risk and be as informed as possible about that risk. So initially, your IRA should contain all that you know about your product, the quality attributes that you have, a reference to any preclinical studies, be it animal studies or new alternative methods, any prior experience with the product class, and how it fits with the theory, right? I mean, just use the literature, tell us what you know. You have to educate the reviewer. You're the experts on your product. Educate the reviewer that's going to look at it. They are not the experts on your product. They have other expertise. Later, the ISI will also contain the assay reports and links to the clinical results and so forth. Now, let's turn our view to follow-on products. We all know that the markets are pretty hungry for these drugs, right? Whether it's peptides, biologics, or nucleic acids, the curve is pretty impressive as to how they're growing in the market and what the expectations are. So follow-ons are going to play a big role. Now, what do follow-ons mean at the FDA? For small molecules, a follow-on is called a generic. It goes through something called the 505J path. And if approved, it can be switched at the pharmacy. You can get the original or a copy at the pharmacy, the generic, it's fine. You pretty much, you don't make that decision, the pharmacy makes it for you. For biologics, they're called biosimilars. And that's, unless they've done some special testing, they are not interchangeable at the pharmacy. For peptides, they can go either the generic route or what's called the 505(b)(2) route. For the generic route, they end up being, again, automatically interchangeable at the pharmacy. For the 505(b)(2), they have their own name, and they have to be prescribed by name by the physician in the United States. So the piece of the pie, or the piece of the number of patients, is going to be quite different. But also the amount of data that you need to rely on. So in a 505(b), for a generic, you need to be sort of the same, right? The standard is that the product has to be exactly the same. For a biosimilar, it has to be highly similar. For 505(b)(2), it pretty much has to be the same, but it relies on some data, but it doesn't rely on some data. It really is, there's all sorts of grays in between. So, again, generic has an identical primary amino acid sequence, right? Exact molecular weight, Q1, Q2, so that means that the formulation is exactly the same in both quantity and quality, right? Consistent higher order structure, if there is, and comparable impurity profile, although that may be evolving right now. So, and then there is absence of unexpected modifications of degradants. That is the standard. It's a very high standard, right? You can ask here, you have to demonstrate the product is the same. For biosimilars, there have to be no clinically meaningful difference. That's a completely different standard, right? Because there is an expectation that the product is going to be pretty much the same, but there's no expectation that the product or process impurities are going to match. You just have to understand their risk. Now, why is that? Why is that difference? Well, because with synthetic peptides and ODNs, it's a synthetic synthesis. You can sort of pretty much, or we hope that you can have a high control over your process. You will understand the structure is well defined. They're small. You can actually characterize the heck out of them, so you really can define something to be the same or not. With recombinants, that's much harder. With a biologic, it goes into cells, you look at the cells differently, and you know that they grow differently, right? We've all been there. So, and also they're structurally much more complex. So, proving that something is the same is almost impossible. Right? Now, if you look at the characterization, and this is critical, before you even think about the immunogenicity part of it, the critical part for any follow-on is that you're going to have to demonstrate that products are same or similar. And that's done using bioanalytical characterizations that are comparative, and you go and you demonstrate, we made product A is exactly the same as A prime, and this is how we prove it, right? All the things having to do with physical chemical properties, sequence, structure, and so forth. So that's pretty much the same. But again, you have other issues with the biosimilars. You have glycosylation issues, you have bioactivity. There are additional requirements to try to establish that we're talking about the same, right? Now, because with a generic, you're demonstrating that the product is the same, there is no clinical trials for safety or immunogenicity. You may get a little one for bioavailability for some products, particularly if they're oral. But there are no clinical trials. If you need clinical trials, you cannot be a J. For biosimilars, because it is assumed that you will have differences, a comparative clinical trial is usually required. Right? So how does that look? The idea is to understand what the incidence and titer of binding antibodies and neutralizing antibodies is in comparison to the RLD. You have to do, it's a parallel arm study. You cannot compare it to what's on the label. You can never match the label. The assays are different. You can't do that. You have to do a parallel arm study. And the aim is to show that there's no difference either in the rate, on the onset, on the offset, and so forth. The problem is that these biosimilars are supposed to have shorter, smaller studies. So doing a short study, it may reduce some of the cost and reduce some of the time, but it means usually that you only have power to assess catastrophic differences. And for many products, the ADA, the antibodies, emerge after months or sometimes years of use. So you can't look at that in a clinical trial. So when you do your IRA, your immunogenicity risk assessment, you need to gear it to explain to the agency what kind of trial you want to do, right? Is your plan to exclude catastrophic impact of safety or exclude a pre-specified difference in neutralizing antibodies or in binding antibodies? You need to sort of understand what your aim is of that study to understand how big that study is going to be. And my recommendation, and I say this because I've seen a lot of companies that go for generics come to the agency with a complete package. We've done this, we're ready to go, please license us. My recommendation is if you're trying to go for a biosimilar and you need to do this, talk to the agency and get their buy-in before. Otherwise, it can be a very painful exercise. So, at some point, the thought was, well, can we do better than clinical studies? And we started with the peptides. And the idea was that if risk is equals the probability times the consequences, and we know what the population and treatment, because that's the same as with the registered drug, and we know what the safety and efficacy of that registered drug is. The only really thing that we don't know here is about the product. And if you've demonstrated safeness, then the only uncertainty has to do with the impurities. And those impurities could be analyzed by clinical studies, but really clinical studies are not geared to look at impurities. They're not good at that. But instead, you could use non-clinical studies. So, and we talked about these non-clinical studies yesterday, so I'm not going to repeat myself, right? The ones looking at MHC and T cells and innate immune responses, and they've been talked about enough, so I'm not going to go into that. But when you're trying to design a molecule and you're using these assays to assess what the risk is, you're trying to be predictive. You're going to say, well, if I make this product, it's going to be maybe immunogenic in 5% of the population. That's my target, right? Here you're using them for something different. Here you're using these assays to say, not what the clinical immunogenicity risk is, but can I support a risk of relatively no increased risk? So can I show that my impurities are not going to modify the risk of the product, which is a very different standard. All of that is in the guidance. It was originally intended for 5 products. It expanded to a lot more products. There's about 20 products now that have been licensed. It's been a very successful program. So, what goes into this IRA for follow-on products? What is the clinical? You want to describe what the clinical experience has been with the reference drug, right? You want to describe a little bit your manufacturing process, enough so that the reviewer that gets your immunogenicity assessment understands how you're making this product and where the potential impurities would come from. You want to talk or give a summary of what the product characterization and sameness demonstration was, or at least put a link to it in your submission. And then you want to really go into the impurity characterization and give your results of any in silico, in vitro, anything you've done to sort of support that absence of increased risk due to the impurities. So, just to sort of go over it again, if you're doing a new product, whether it's under BLA or NDA, you will need to assess the immunogenicity, you'll need clinical studies to look at incidence and impact, right? If you have follow-on products, you have a biosimilar, right? You will have to do your comparative and analytical assessment, and then followed by clinical studies. If you have synthetic peptide, it could go via the 505(b)(2) path or the generic path. And in both of those, you can do just a comparative, analytical, and then depending on the risk, end up doing some additional in vitro studies. Now, remember that these will only inform you about incidence and not about clinical impact. Now, the interesting thing is that until now, and I would say now as in the last month, recombinant peptides had to go mostly through clinical studies and were not allowed to be in the generic space. That may change as we speak. There is an opening for maybe introducing recombinant peptides as generics, which I would assume will mean a lot more reliance on these type of in vitro studies. So something to look for. And the expectation is that if this works, right, if it can go through that kind of assessment, then eventually for biosimilars, the clinical studies can either be replaced or supplemented by non-clinical studies. So that's the hope, that's the intent. So do all products need such an extensive immunogenicity risk assessment? No. So really it is a sliding scale and it depends on the risk. And whether the product is, what kind of risk the product has, from an oral synthetic all the way to a non-redundant known factor that can cause deficiency. There's a big range, and how much you need to do really depends on the risk. So once again, that IRA becomes critical. So I'm going to go, I went yesterday over this, so I'm not going to repeat myself because I only have 3 minutes left. But I did want to mention some pros and some cons of how the reviewers see these assays. So in silico, there's multiple tools by multiple providers. So that's a big advantage. High throughput, low cost. You can cover multiple MHCs, although you should be aware of how deep the knowledge is for the different types of MHCs, as was pointed out yesterday. And the potential impact of impurities can sometimes be used assessed using this. However, there's weak assessment of unnatural amino acid side chain modifications in cyclic peptides. Most of the studies are based on HLA-DR, but the PDQ are starting to gain some traction. And one of the big issues is susceptibility to informational bias due to underrepresentative training or flawed assumptions. That's a big issue. And so, careful, look at the data, but look at it critically. And this is one of the problems that it requires the regulatory agency to understand what you're doing. Now, you as a sponsor don't have access to the algorithms. So that becomes a little bit complicated, right? Because the person who's giving you the service to look doesn't always want to share that proprietary information with you. So some companies will establish a master file with the FDA, so something to look for. And then the last thing that I want to point on is on data interpretation, right? So you've done your study, and all of a sudden you find that there's this one MHC where this impurity could create a problem. So it is important to take a step back and think, okay, how does that really impact on my risk? And how do I compare it with data from a clinical study where MHC was not really assessed, right? So there's it's not a, give me a little number and I will understand what the risk is. You have to put it all in context. We've talked today about MAPS and in vitro MHC assays. They're very powerful tools. They're also pretty involved, particularly the MAPS assay, right? So it's low throughput, it's complex. Few companies do it on a regular basis. So if you're going to do it, you want to justify what method you use, what the cell culture conditions were. Please be explicit about what the MS settings are and the data analysis and algorithms used, because that's one of the things that we see that become a problem. Sorry. And if you're doing an in vitro MHC binding, an important issue is to consider what is the affinity of the peptides you're trying to displace. That's something that has trumped up some submissions. With regards to T-cell activation assays, I'm just going to go through the limitations just because of time. Be aware that naive T-cells, if you need to look at those responses, are in very low amounts, and so you need to consider that when you're designing your study. They're complex, there's high donor variability, and one of the big short problems that there is currently is that there's as many methods as providers and few suitability standards that are used by all. So that's something that has, again, trumped several submissions. With regards to the innate immune activation, we went over it yesterday. There's different formats, right? But we talked about the low frequency of APCs in peripheral blood, the high donor-to-donor variability, the lack of universal standards is a problem, and the potential interference. So again, do it carefully. Before I finish, I do want to spend a minute on nucleic acids, because I think that's a field that's sort of getting traction, but coming from behind. So there's about 23 FDA-approved oligonucleotide drugs right now. There are no generics in the market. None have been approved. The number of impurities is astronomical, and it's pretty much impossible to separate them and to individualize them. So there is a different thinking around what to assess. There's very little clinical experience, so right now there's pretty much no guidance. We understand that, and we tell all the FDA says, not we, but all the FDA says anymore is just come talk to us. You should know that what concerns them is immunogenicity, inflammation, complement activation, and thrombocytopenia. Those are the four things. Of those, immunogenicity is probably in the lower rank, right? But these three are really critical. So far, there are really few companies that have assays geared toward nucleic acids. There's very few published. We have one paper submitted, one that has been published, but there's very few really out there regarding the assessment of nucleic acids. So to summarize, immunogenicity can reduce the safety and effectiveness of valuable therapeutics. I don't know if it uses them to tap water, but yeah. But it is really problematic. So identifying the risk factors and mitigating them can really accelerate your development and accelerate licensing. You can use the in vitro and in silico tools to inform the risk and understand it. And be aware that the new regulatory paths may rely even more heavily on these types of assays. So this is one of the things that I mentioned yesterday when Tim asked us to look into the future. We're here, right? We're trying to get here to understand what the local systemic immune activation is. What we need to get is here. How is that going to, how can we really predict what is going to cause clinical damage? So the current focus is to rule out increased risk and the risk of inducing ADA to different epitopes that could spread. But the aspiration is to get to the clinical implications. With that, I want to thank you. Just remember to understand the product, identify the risk, and mitigate the uncertainties. Thank you. Questions? Yes.
Well, nice talk introduction. I think for small nucleic acid drug is also quite popular globally and also in Chinese new drug development. Also, previously when we trying to design the drug, I think we always want to add non-natural modifications like phosphathionic bonds or even like I think for nucleic acid, also for peptides, because it's chemically synthesized, we all intend to adding non-natural building blocks to improve the stability. But we also know that adding these things, it also compromise the treatment, especially in vivo. But also like from drug development, we only look at the curing effect or knockdown effect. We never really assess the immunogenicity. So is there any already some like data sets can simulate the effect of those standard modifications? Because there's always a trade-off like stability, activity, and immunogenicity.
You're talking about oligonucleotides, right?
Yeah, oligonucleotides.
Well, I think there's a number of papers that look at some types of modifications like phosphorothioates and some of the usual modifications. Yeah, there's a number of papers that have looked at those and tried to assess, particularly with phosphorothioate. And they have a potential impact on, they are pro-inflammatory, they can be pro-inflammatory sometimes. But again, it's a nascent field, only 23 products approved, most of them for very small indications. Most of the clinical trials have maybe 60 patients, 100 patients, because they are for rare indications. So I think that in the next couple of years, we're going to see a number of products approved that have had thousands of patients. And I think that's going to be very informative. It's pretty early days in the field, incredibly.
Thank you.
Hey, Daniela, thanks. Wonderful, comprehensive. It gave me a total summarized picture of what we are intend to do. One question I think I was discussing with Wenzel also yesterday. What is your thoughts on the IRMIs being considered for the oligonucleotides versus the peptides?
Yeah. So from what I've seen in the lab, because we've worked with oligonucleotides, and what I've seen a little bit in the regulatory space, one of the main concerns is that oligonucleotides can sometimes synergize with some other PRRs or pathogen recognition receptors, ligands for pathogen recognition receptors, right? So that is a concern, that you have a low level of activation, but it can be synergized because it's an oligonucleotide. There's a good paper by Pollock's group, and they show that a non-TLR9 triggering impurity could actually synergize with an oligonucleotide and dramatically increase the innate immune activation caused by TLR9. And we showed it between LPS and a TLR9 agonist as well. So you have to think about those potential interactions, and that's why I think that an assay like the IRMI might be useful. On the other hand, what we've seen is that a lot of the APIs can release, dampen the response of the immune cells. So hopefully, we've submitted the paper, hopefully it'll come out soon. But there's really, you have to really work with that balance, because the API can be really dampening of the signal.